Hi, everyone. Our presentation is on genetic polymorphisms of the renin-angiotensin-aldosterone system in end-stage renal disease, presented by Athena Braille, Marissa Mall, Tori Simmons, and Sydney Kaler.
So the hypothesis for this study is that specific single nucleotide polymorphisms in the AGT, ACE, and CYP11B2 genes. Those specific variants are M235T in AGT, an insertion deletion variant on intron 16 on ACE, and 344CT on the CYP11B2 gene. And the hypothesis is that these variants are more prevalent in end-stage renal disease patients, and that the presence of them are associated with a more rapid progression of end-stage renal disease patients as well. There have not already been any studies on how these polymorphisms relate to end-stage renal disease, but there have been previous studies that have identified these specific variants to have an association with an increased prevalence in hypertension. So those previous studies inform the hypothesis for this study because hypertension is a contributor to renal failure or the development of end-stage renal disease. And it's a risk factor. So as renal function decreases, hypertension prevalence increases. Genetic susceptibility to hypertension may predispose individuals to developing renal disease and eventually progressing to end-stage renal disease. And genes of interest when observing hypertension are genes that regulate renal sodium absorption, which is also called the renin-angiotensin-aldosterone system. And those specific genes that we will be discussing are angiotensinogen, or AGT, angiotensin converting enzyme, or ACE, and aldosterone synthase, or CYP11B2. So the first gene of interest is angiotensinogen, like I mentioned, and this is a protein coding gene. It encodes for the protein pre-angiotensinogen, which is cleaved by renin into angiotensin-1 in response to lowered blood pressure. This protein is responsible and involved in maintaining blood pressure and body fluid and electrolyte homeostasis. And it's also involved in the pathogenesis of essential hypertension and also preeclampsia. Mutations in this gene can be associated with an increased susceptibility to essential hypertension, and the variant of interest in this study, M235T, has been observed to be in higher presence in people with hypertension, and also an increase in that variant has resulted in an increase in severity of the hypertension. And it's also associated with a higher plasma level of AGT in those subjects with hypertension. The next gene involved in the study is angiotensin-1 converting enzyme, or ACE. It is also a protein coding gene. It encodes for an enzyme that catalyzes the conversion of angiotensin-1 into the physiologically active form of angiotensin-2. This enzyme also activates the vasodilator protein bradykinin. So it is involved in maintaining, it generally increases blood pressure and maintains body fluid and electrolyte homeostasis as well, just like AGT did. Mutations in this gene are associated with cardiovascular pathophysiologies such as hypertension and renal disease. The specific variant of interest in this study is an insertion deletion on the intron 16. The deletion of this polymorphism has been associated with higher plasma ACE activity than subjects that have the insertion allele instead. And the last gene of interest in the study is aldosterone synthase gene, or CYP11B2. It encodes for cytochrome 450 monooxygenase, and that is an enzyme that catalyzes the biosynthesis of renin-angiotensin-aldosterone, which is also involved in homeostatic regulation of blood pressure and also helps control plasma sodium and potassium levels, which are important when considering renal function. Variant of interest here is a single nucleotide polymorphism at the -344 position on the gene promoter. And it has been discovered in 2 separate hypertensive populations. And it also has been seen to be associated with an increased ventricular size of the heart and impaired diastolic function in blood pressure.
So this is a clinical trial, and it's important to note it wasn't possible for it to be a randomized controlled trial for intervention and control because it was based on disease state. The study subjects were 260 white patients with end-stage renal disease, either with dialysis or transplant. The control group was made up of 327 white control subjects without renal disease, and genetic analysis for the M235T AGT and deletion and insertion ACE and -344 CTCYP11B2 genotypes were performed in all subjects, both control and study, and compared to determine susceptibility to renal disease and progression of renal function loss. This was done by isolating peripheral blood using the nucleon BA-CC3 DNA extraction kit. Then deletion of polymorphisms was performed by polymerase chain reaction analysis using primers. So this table shows the baseline demographics from the material and methods. The DNA was prepared by isolating the genomic DNA from peripheral blood. The DNA was analyzed by polymerase chain reaction to detect polymorphisms. Polymorphisms AGT and CYP11B2 were analyzed using restriction typing, while amplification of intron 16 was used for ACE analysis. Then PCR products were analyzed on gels using a 2-buffer system. Table 2 shows the primers utilized for each polymorphisms. T-test and ANOVA were used for statistical differences between means of continuous variables, while Wilcoxon or Kruskal-Wallis tests were used for variables that were not normally distributed. The 2x2 contingency table X2 test was used for categorical values, and Hardy-Weinberg equation was used for expected disease frequency for the target population. All of the previously listed statistical analysis were performed using the SysStat 8.0 SPSS software.
Okay, so now for the results of the study, the genotype frequencies did not differ significantly between the end-stage renal disease and the control group, but they found that when they compared the end-stage renal disease diabetic patients to diabetic patients without neuropathy, the prevalence of the AGT-MN genotype was lower. So you can see that here, while the TT genotype was higher. And so when you're looking at this graph, the black is the diabetic end-stage renal disease, and the white bars are patients with diabetes mellitus without neuropathy. So basically the graph is showing that patients with diabetic end-stage renal disease were more likely to have the AGT genotype TT and TM, where the MM genotype was more likely in the patients with diabetes mellitus without neuropathy. So it's this bar right here. And then we'll talk about this in the next slide, but the total end-stage renal disease population, the progression was faster with the ACE-DD genotype when it was compared to the DI and the II alleles. So when you look at the left graph, figure A, this is showing the ACE genotype and the rate of progression of renal disease in years. from the time of diagnosis to the onset of end-stage renal disease in the entire end-stage renal disease population. Graph over here, B, shows the interaction between the AGT and the ACE gene polymorphisms on the rate of loss of renal function for the end-stage renal patients homozygous for the AGT-MM gene. And then looking at this graph on the right side, it shows the genotype and the rate of progression of renal disease in years from the time of diagnosis to the onset of end-stage renal disease with glomerulonephritis. And it was found for the patients with glomerulonephritis of any etiology that there was a significant correlation between the rate of progression of renal failure and the AGT genotype. So you can see the genotype for TT, the progression was fastest in around like 9 years. And then for the MM genotype, it was a little bit slower at 12 years.
So there are several points of discussion to note from the results of this study. First, the overall likelihood of end-stage renal disease and faster progression of the end-stage renal disease is linked with the AGT genotype in diabetic patients. Furthermore, faster progression to end-stage renal disease is associated with the ACE genotype when the total population with end-stage renal disease and the AGT genotype, or when patients with glomerulonephritis are considered. There is no association found in this population between the CYP11B2 genotype and hypertension in the patients who did not experience renal failure or rate of progression of renal disease in end-stage renal disease population. Because of this finding, we can see that the 344CT CYP11B2 genotype would not be a primary candidate for susceptibility or progression of renal disease. The ACE-DD homozygote renal patients showed faster progression to end-stage renal disease than the ACE-DI or II participants. So the ACE-DD genotype is found to predict quick progression to end-stage renal disease in AGT-MM homozygotes, and the AGT-MM homozygotes only had a more rapid progression when the ACE-DD genotype was present. Unlike AGTT, which that allele shows the most rapid progression, which we just saw in the results, regardless of the ACE genotype. Progression to end-stage renal disease was comparable among men and women with the DD genotype, but was shorter in men who had the II or ID genotype. And we'll touch on this in the limitations, but this could suggest a limited effect of ACE inhibitor therapy. Patients with glomerulonephritis of any etiology, which we also just touched on this in the results, had a significant correlation between the rate of the progression of their renal failure and the AGT genotype. And then once again, as we just mentioned, the AGTT showed the fastest progression overall, about 9 years, compared to the other two alleles. And overall, there was no association between the age of onset of the end-stage renal disease, the etiology of renal disease, the presence of hypertension, and the number of drugs, the anti-hypertension drugs used to control their blood pressure, and the AGT, ACE, and CYP11B2 gene polymorphism. So these findings indicate that the ACE gene polymorphism is at some level a predictor of progressive renal function loss in patients with renal disease. In patients with glomerulonephritis who are homozygous for the AGT-MM genotype, there seems to be a greater influence of the ACE genotype. So some of the limitations of this study were that the selection bias was based on ACE genotype, determining cardiovascular mortality. And in a study population with renal disease, this increased mortality before their end-stage renal disease occurred, which would lead to underestimation of alleles that promote renal function loss. The second limitation, which I briefly touched on in the previous slide, was that the ACE inhibitor therapy could possibly be a confounding factor based on the progression to end-stage renal disease that was comparable in those with the DD genotype, but seemed to be shorter in men with the II or ID genotype. Some of the strengths of this study were that it had a large end-stage renal disease patient population from a single center and a large amount of control subjects, which was 327. The population is also considered to be homogeneous because more than 88% of the participants were from the Swiss-German area. And finally, the last strength was that the phenotype for renal disease was defined strictly. So there was only 16% of the patients who suffered from a renal condition that was deemed to be unclassifiable. So as far as further research goes, the functional relevance of AGT gene polymorphisms and CYP11B2 polymorphisms is a topic that has been under exploration from several different groups and researchers. With many studies that show differing or contradicting results, these RAS system genes appear to be the candidates genes for exploring and gaining understanding of all the inter-individual differences in the development and progression of end-stage renal disease. And these are our references. Thank you so much for watching.